So before we start, make sure that you've taken the 114 research sheet and pasted it into your notebook. And what we're going to do now is analyze the blood that was found at the crime scene. We can't assume that was Anna's blood, so we have to do some blood typing. So that's what we're going to learn about. So you can fill out this resource sheet as we go through these notes. So the first thing we're going to have to know is what antigens and antibodies are, because that's how we type blood. And then we're going to learn about how they're used to type blood with something called agglutination. So first, what are antigens and antibodies? So antigens are little signs outside of the blood cells that tell them what they are. So we have four major blood types. We have type O, type B, type A, and type AB. Type O has no antigens. You know how it has no sign. So type O could really be called type 0. There's 0 antigens on the outside of it. Type B has B antigens, and you notice that they're blue, and they are diamonds, and then type A are yellow circles. So each antigen is different and specific. So you'll only find A antigens on type A blood and only find B antigens on type B blood, and they're on the blood cells. And then finally, AB is actually both combined. They have both of those antigens on them. And it's easy to remember that because it just goes with their namesake. So go ahead and fill in that. You can fill in one of these drawings as an example, an antigen, and put in your antigen part. So now what are antibodies? Antibodies are things to attach to antigens and mark them for the immune system to destroy them. All right, so each antibody is specific to its antigen, because think about it, if you had your antibody for your own antigens, you would constantly be attacking yourself, and that causes diseases. There's actually something called that, but we'll talk about that later. So type A has actually type B antibodies. You don't have to worry about this right now, but notice how they won't not fit together. All right, we'll talk about how the antibodies and antigens actually interact, which is down here, is when the B antibody comes into touch with a B antigen, they actually attach to each other and causes the blood to clump together. All right, so there's B antibodies and there's A antibodies. Type A has no antibodies in them because otherwise the blood would be attacking itself. And again, these antibodies are just floating around the bloodstream. They're not actually attached to blood cells. They're floating around the bloodstream, and they're actually looking for other blood types or other things that shouldn't be in the body. So antibodies, you should put that in that definition, that they're looking for antigens to destroy them that shouldn't be in the body, and you can put down a drawing for that. Now, the last thing is something called agglutination. Agglutination is just a fancy word for clumping. That's when the blood clumps together. So, in this example down here, there were B antigens on the cells. So, when in the presence of B antibodies, they fit together and they clump. Over here, in this A bottle, there were A antibodies in here. Now, you can't see a picture of this, but the A antibodies probably had a different shape, so they would not fit with the B antigen. The A antibodies would only fit with the A antigen. Okay, so if the A antigen was present, it would clump. If it wasn't, it wouldn't clump. So in this case, it did clump because B antibodies were clumped. And then over here, if we would have shown it, it probably would have clumped. But anyway, B antibodies will cause any blood cell with B antigens to clump. A antibodies will cause anything with A antibodies to clump or agglutinate. And we'll talk about that in a little bit. But right now, let's just get those terms down. So do you understand what antigens are? Do you understand what antibodies are? And do you understand what agglutination is? So make sure you talk with your partner and have a facilitator, have everyone chime in and make sure they can specifically define and understand what an antigen and antibody is and how they interact for agglutination. All right, now, how does this agglutination factor help us? So you can look through this PowerPoint, but I'm going to help you with this little chart. It's not too hard. It's actually super easy. So if we have, you have type A first. All right, and we're going to mix type A with a antibody. So what happened is 8 antibodies, and type A, remember, has the A antigen. If you want to write that next to your table, you can. So therefore, they match, and it would cause it to clump, which means we'll put a little plus sign. With antibody B antibody, remember the B antibody is a different shape than the A antigen. And this one, type A, has no B antigens. It only has the A antigen. Therefore, it will not clump. It will be minus. So, therefore, type A should only clump in A antibody. Type B, remember, only has the B antigen. Therefore, it's the opposite. It will not clump in A antibody because it has no A antigens for the antibody to attach to, but it will clump in the B antibody. Type AB has both antigens. Therefore, it should clump in A antibody because it has the A antigen, and it will clump in the B antibody because it has the B antigen. Finally, type O has none. Therefore, it will not agglutinate or clump in either one. All right, so try those practice ones below. And so pause the video and try those practice ones below. All right, now here are the answers. So if you haven't tried them yet, the first one, that little plus sign there, the first one on the left would be type A because it only clumped in type A. The second one would be type B because it only clumped in type B. And the third one would be type O because it did not clump in either one. All right, so what you should get is that it only clumps in the present of it. And the easy way to remember this is if it clumps in A, it's type A. If it clumps in B, it's type B. If it clumps in A and B, it's type AB. And if it doesn't clump in any, it's O. So, right here, I just want to show you, this is eventually what it's going to look like. Clumping looks like this. There's little dots here. Not clumping, it will look like it's just a red spot. So go ahead and go through success criteria 2. Do you understand the blood types and how we identify blood? Because in a second, you're going to be doing a lab, and I'll talk about that just real quick. Okay, so now doing the lab. The directions are pretty clear. You put a clump of, let's say, the blood found in the scene here, a drop of blood found in the scene here, and then you add antibody A here, antibody B here, and then you mix it up, and mix it up real good for like 30 seconds. The other key thing that will help you is put a paper with writing underneath. Because sometimes it'll be hard to see the agglutination unless you have writing underneath, because then it will be hard to read. So it will agglutinate. I probably spelled that wrong. It will agglutinate when you can't read the paper. All right, and you guys can always verify your blood types. Once you blood typed everybody, you can just verify your blood type with it. And your goal is to try to figure out whose blood might have been at the scene. You might not be able to narrow it down to one person, but you should be able to cross some people's blood out. All right, that is it. Oh, make sure you clean up your supplies when you're done. Wash off your tubes and stuff.